Hello and welcome. Here we are once again. It's NEUR3301, my friends. We're just waking up on a nice cold frosty morning and enjoying our experience of sitting down and talking about some genetics. So today's topic is going to be the subject of mood disorders. We'll jump right in. So actually, well, we won't really jump it right in because before we talk about the subject, I have a few aspects of course homeostasis, I guess you could say, administration. So the first is that you should have received an email explaining that the third assignment is now available. It's the last one you have to do. It's all about ChIP-seq analysis or chromatin immunoprecipitation analysis. So this will give you a sort of yet another type of, or yet another, I guess, modality or form that genomic data can take. You've seen your RNA-seq data, you've learned how to use the genome browser, and now you'll learn how to work with genomic regions. And that's usually the way that epigenetic information is conveyed. So with these three assignments under your belt, you should have at least some starting point for working in genomics and epigenomics. Certainly if you were to go into it, sort of as a researcher or in a more professional context, you would have to sort of relearn a lot of these skills using maybe more appropriate software and methods. But I think that what these should give you is a sense of the questions that you need to ask, the way the data is structured, and the basic sort of steps you would go through to do things. You can learn to use fancier tools to do it, but fundamentally what you're doing is the same thing. So hopefully there'll be a value to you. I want to specify, and I hope that I made this clear in the announcement that I sent out, the motif enrichment tool, C, that's in the assignment. That is a web-based tool that takes quite a while to run. It could take up to 24 hours to run from the time you put in your data to the time that you get results. For this reason, it's essential that you start your assignment at least a day before it's due. It would be very unfortunate if you waited until the last minute to do your assignment and it's 11:57 the night of the deadline and you're waiting for this thing to produce results. That's just not going to be the way that that's not the right way to do things. So I'm warning you now that you should try to get this started a day or two before the deadline is before it's due, in order to have enough time to let this package run. So watch out for that. We're going to talk about the results of the midterm exam in the next slide. Hopefully this result doesn't give you a mood disorder yourself. So we'll start by thanking the TAs, Priya and Sarah, in this class for their hard work marking and getting us a quick turnaround of just a little bit more than a week's time. I think that's it's pretty nice to have a quick turnaround for your exams, because the what value do they have if they can't influence your practice? So I'm happy to say that people did a fair bit better on this one than the first one, which is roughly what I was expecting. So here the class average was 78%, 78.47%. The median was 76%, meaning that half the people got better than 76%, and the standard deviation was 16.35. So here's a histogram showing the distribution of grades. You can see that it's very skewed toward the positive end. This is partly because of how I scale the grades after I get them, to sort of, I guess, kind of boost the marks a little bit, just from the sort of normalization that I tend to do on the grades. But it's also just because people straight up did do better on this exam than the first one. Your marks, by the way, I haven't posted them on Brightspace yet, but they should be available later today. You'll be able to see your own individual mark. But I'll just go over some of the class parameters, because I do love analyzing things. Some other things that we can look at is the relationship between your exam score here on the y-axis and the amount of time it took people to write the exam. So on the x-axis, we have time to complete the exam as a % of the allowed time. So if for example, you had 90 minutes to write the exam and you took all 90 minutes, then your score, or your value here, would be 100. And if you only took half as much time, you'd be down here. And so this is a rather interesting thing, because if you recall from the first midterm, the relationship between writing duration and grade was actually the opposite in the first midterm. It was a negative correlation, whereas here it's a positive correlation, meaning that on average, the longer it took you to write the exam, the higher your grade was. Now, I think this is not really something you could interpret that meaningfully, because almost everyone took the full amount of time. In fact, we could sort of see that, many of you seemed to have run out of time toward the end of the exam, even though there was actually only 18 questions here, so there was 2 fewer than the last one. But clearly the timing is a problem. I think it's a bit of a nuisance with these online Brightspace-based quizzes. The fact that I'd like to know why my webcam is flashing and flashing on and off. That's quite irritating. Not flashing on and off, but the light adjustment is really causing a lot of problems. Anyway, it just has to do with where I have my head. I guess maybe it doesn't like the window open behind me. Hard to say. I hope that's not too annoying for everybody. I'm trying not to look at it, but it probably would be pretty irritating to watch. Anyhow, I don't know how to fix it. So most everyone took the full amount of time or very close to it. So that's a bit tough with these online things. On a paper-based exam, you would be able to look over the whole test pretty easily and decide where you want to allocate your time. With this format, the questions are randomized in their order as a security measure, and of course, you might end up with the longest question at the very end, and that's really not ideal. So I don't really know what to do about that. But I guess that's just kind of how it works. Brightspace just makes it that way. So, we'll see. And then over here we have your exam score plotted against how long you waited before starting. So if your start delay was 0 hours, it means you started the exam at 8 a.m. on Monday. And if it was 24 hours, it means you started the exam basically at the last possible minute. And you can see here that there's a bit of a negative correlation. So people who waited longer to start the exam didn't tend to do as well as people who started earlier. Maybe this also has to do with just the time of day. You can kind of see nighttime, the time of late night where nobody's really writing, and then early morning, the morning that the exam is due. Not a great time to be writing, but who am I to judge? Everyone's got their own schedules. And then we can look at the rate of improvement or de-improvement, I guess, the opposite of improvement. So here we have the score on the exam plotted on the y-axis, and then we have your scores on quiz 1 and quiz 2, and each individual person's points are connected with a line. So you can see for somebody like this, their score went up. For somebody like this, their score went down, and the overall tendency for most people is an improvement. Some people did go down, but they were in the minority. Here's a histogram showing the same sort of thing. On the x-axis of the histogram, we have the change in percent from midterm 1 to 2. So anybody on the positive end of this had their mark go up on the second midterm, and anybody on the negative side had their mark go down. And then the bars are simply colored by the percent change to give it a bit more visual flair. So you can see many people went up by 10 to 15 percentage points compared to the first exam, which I think is quite a nice improvement. And I don't think it's really because the exam was any easier. Maybe it was because I did a better job of preparing you for it, or maybe you have all just sort of learned kind of the way this course works, or maybe the material in the second half of the course is a bit easier. It's kind of hard to say, but at any rate, we love to see the improvement, folks. We love to see it. So now I want to just go through some of the questions that people had particular difficulties with on the exam. So here I've made a heat map that's been hierarchically clustered. Each, in this case, column is a student, and each row is a question. And then I've used k-means clustering to break them up into 2 separate groups. So you can sort of see that here there are sort of 2 main clusters of students. The students who did generally quite well, and most of their questions were sort of toward the higher end of the spectrum, and then there's the cluster of students who didn't do quite as well. And then there's really nothing, there's no real texture to the data. They just tended to not do very well on the written portions or anything that was marked on a continuous scale. So there's not a huge amount that we can learn from this, but it is sort of neat to see how things break down. You can see, like, for all these ones that are all pretty much red, those would be like the multiple choice questions that almost everyone got right. There's not a lot to talk about there. Question 10 is an interesting one. I'll get into that one, where surprisingly many people did get this one wrong. Many people, of course, got it right. More people got it right than wrong. But a surprising number of people who did very well in the exam overall ended up getting question 10 wrong. So we'll talk about that in a little bit. And let's look at how the final grades will be allocated. So I thought it would be nice to, I guess, partly incentivize better studying habits, perhaps, or like, trying to look over the whole course and build like an integrative, cumulative understanding of our material. To incentivize this, I can have a sort of a reallocation of the final grade. So my thinking is, I'll offer the opportunity to reweight your grades such that the final exam can replace one of your midterms. So the idea is that, here it's set up so that your first midterm is worth 20, your second is worth 20, and your final is worth 30. If it would happen, like if it would improve your grades, I will reweight your overall class or your overall grade such that the final becomes worth 50%, and then we drop your lowest midterm, which for most of you would be midterm 1. For some of you it would be midterm 2, but for the majority of you it would be midterm 1. So if your grade is improved by doing this, then that's the arrangement that you'll be given. If it doesn't, say for example you do worse on your final exam than either of your midterms, then your grade weighting will be kept the same. So you can't really lose here, you can only win. All you have to do is do better on your final exam than you did on your first or second midterm, which I think is probably within reach, but it won't be, it's not going to be a freebie. You'll still have to do the exam and do really well on it. But I think now that we sort of see a positive trend among within your grades, I think the odds are pretty good that you'll do well. I shouldn't say, I don't know how you're going to do on the final exam, but if we can look at the delta between midterm 1 and midterm 2, it's a positive trend. So we would hope that if you maintain your same level of dedication and your same sort of level of understanding of the material, then things should also improve for your final. So if that's the case, that'll be good for your grades. Hopefully that's clear to everybody. It's not too weird and crazy of a system, but it's something that I can add pretty easily. Okay, so before we get into the topics for the actual lecture today, which I think, the lecture on mood disorders we don't really have a huge amount to say, to be honest. There's mood disorders aren't an area where the genetics are particularly interesting, but we can look at some of the questions on the exam and try to just review them as to help anybody who maybe didn't do as well. The first question, and so I'm just going to go through some of the questions that had the lowest marks or the highest level of variability. There's no point in going over questions that almost everyone got right or questions where the average was quite high. These are ones where the performance was rather more variable and people didn't tend to always do quite as well. So this is question 10, that one that I thought had a very strange kind of dichotomous pattern. And the question goes as follows: which of the following best describes a methylation quantitative trait locus, an MQTL? And the correct answer is when SNP genotype affects methylation level at a nearby CPG. And there are a couple of slides where this is discussed, but one that, so there's one lecture where we talk about it. It was the one where I sort of showed that iceberg meme, where there's deeper levels of ways in which genetics can affect molecular biology. But the area where you could have found the answer to this would have been on this slide from the schizophrenia lecture. And what it tells you is that most of the variation in DNA methylation that you see comes from cell type or tissue type. An important fraction also appears to be under genetic control. And so MQTLs have an effect on methylation. That's plotted here on the y-axis of this figure, and they tend to be fairly close to their respective SNP. So the SNP distance between, or the distance between the variant and the CpG is given on the x-axis. So if the distance is 0, they're right on top of each other, and you can go away up to 500 kb or half a megabase. So the answer is when SNP genotype affects methylation level at a nearby CpG. These other ones, I guess, maybe had sort of a tempting character to them because it is true, it's not, it's true, methylation levels do differ substantially between tissues, but that's tissue level variation, not genetic variation. All of your tissues have the same DNA, more or less, so that wouldn't be a type of genetic variation. When DMRs overlap disease-associated SNPs, this also is not the same thing because DMRs exist on their own terms, and if they happen to overlap a SNP, that is informative in a way, but it's not necessarily the case that the SNP causes the DMR. And then finally, when CpG methylation affects the expression of a nearby gene, that is also not an MQTL because M in the M means methylation, so what's being changed is methylation levels, not gene expression. EQTLs are when a genetic variant affects the expression of a nearby gene, but that's also not really what's being talked about here. So that's something to know. The next one is question 14. A number of people had problems with this. The question is, cellular heterogeneity is a concern for which kind of studies? And this was a multi-select question, one of the fun things you can do on the Brightspace platform. And the answers are WGBS, RNA-seq, and ChIP-seq. The answers are not exome sequencing and GWAS. The reason for this is that your DNA is, similar enough across all of your tissues that cellular heterogeneity is not a meaningful problem when you're trying to just measure somebody's genetics. It is true that there is some mosaicism, but that's not taken into account or really worried about in these models because the number of, so for any given variant or for any given SNP or location throughout the genome, the odds of there being enough mosaicism to severely throw off the results of exome sequencing, or especially of a GWAS method, very, very low. Because typically these variants would appear in only a tiny minority of cells and thus wouldn't contribute a whole lot to the variation in the bulk DNA. For GWAS methods that use microarrays, you probably wouldn't see them at all because most of the mosaicism comprises de novo mutations, which of course aren't covered by the GWAS arrays. So the correct answer to this was WGBS, RNA-seq, and ChIP-seq. And we talk about this a couple of places. I think it's kind of one of my recurring themes, this question of cellular heterogeneity. But one slide where you would be able to find it comes from here, which was in the Genetics of Neuroanatomy lecture. But it's, like I say, it's dealt with a few different places. I think it's probably one where there's no slide where I explicitly say this. It's one of those questions where you're meant to think about the implications of what I've told you over multiple lectures and try to put them together into one thing. I still quite like that type of question actually, because I think that really tests whether you're, sort of whether you're getting it or whether you just sort of have a surface level understanding of things. Okay, so here, and I guess you could sort of imagine this is a bit of a contrast to the last question, but the question here is asking, and this is number 5, does every cell in your body contain the exact same DNA sequence? Why or why not? How is this relevant to conditions such as Huntington's disease? So this is given slides 12 through 17 of the Huntington's lecture. We know that mutations can build up in individual cells. This is called somatic mosaicism. So no, every cell in your body does not contain the exact same DNA sequence. They're close enough that they're functionally identical, but there can be variants. Why or why not? This is because mutations can accumulate in cells due to various things. Things like ultraviolet radiation, if it happens to be exposed to sunlight, errors during division can happen, exposure to mutagens, as well as just random chance. So people who have a very difficult and rough life, who are smokers, for example, and flood their lungs with smoke and other kinds of carcinogens, or people who live in, who work in, nuclear plants and handle plutonium all day or whatever. These are all things that can increase the rate of mutation. How is it relevant to conditions such as Huntington's disease, well, the somatic mosaicism is quite relevant to this because repeat expansion of the Huntington's associated CAG repeat is something that can happen in individual cells in the body, somatic repeat expansion, particularly in the striatum. And this actually can explain why the age of onset differs between individuals with the same number, who are born with the same number of repeats. The number of repeats can expand in relevant tissues like the striatum and cause an earlier age of onset. And this is actually pretty relevant because now we have GWAS data showing that things like the MSH3 gene, I don't know why I have so much trouble saying that word, the MS, I don't know, I got to sort of decode my my brain and figure out why I can't say that, but MSH3 gene, which is a DNA repair enzyme, and it's involved, so mutations here or variants here can affect the age of onset. So that's the kind of answer we're looking for there. There's a handful of slides that give you everything you would need to say about that. The next one is explain the concept, this is question 9, explain the concept of genetic correlation and how it can be derived from GWAS studies. What is suggested by the fact that schizophrenia and bipolar disorder have a positive genetic correlation? So this is slide 10 from the part 2 of the schizophrenia lecture. And what I'm basically, this is not slide, this is slide 10 from the ASD lecture, my apologies. So what you're talking about here is that GWAS studies can generate a test statistic of some kind, whether it's an odds ratio or a coefficient, like a regression coefficient, beta value kind of thing, based on allele dose. And then you can compare the direction and strength of those values between phenotypes. So you could plot, for example, schizophrenia against bipolar disorder and see that typically SNPs that or variants that increase your odds of schizophrenia also increase your odds of bipolar disorder, while at the same time variants that decrease your odds of one also decrease your odds of the other. That is the essence of a genetic correlation. The overlap and correlation between schizophrenia and bipolar disorder seems to suggest that they are caused by similar neurological or neurodevelopmental processes. And, we might even go as far as suggesting that there is a common genetic architecture for a wide range of mental illnesses. The next question is 11. This is the last one I'll talk about. If you have any more questions about the exam, please feel free to contact your TA to arrange a review session. I'll instruct them to make their times, set up a time where you can do this. So here's question 11. Explain the role of DNA methylation in the expression of Fragile X syndrome. What role do methyl donors and 1-carbon metabolism play? How is CGG repeat expansion expansion affect DNA methylation? Why does DNA methylation affect the physical structure of the X chromosome in Fragile X syndrome? So these are slides 9 through, this question rather concerns slides 9 through 15 on the Fragile X lecture. So there's quite a lot of slides you would have to take into account. But yeah, so the level of methylation at the CGG repeat is actually the main determiner of the Fragile X status. The mutation on its own doesn't necessarily mean that the Fragile X marker will be expressed and you'll have Fragile X syndrome. It depends on the level of methylation at that repeat site. And they've known for quite a while, based on studies of cell culture, you remember Patricia Howard-Peebles and her work showed that the amount of methyl donors in the culture medium is essential for the expression of the Fragile X chromosome or the Fragile X marker. If you grow cells in media that's depleted for methyl donors, and thus they can't do very much DNA methylation, they don't tend to express this marker chromosome in the same way. They don't tend to have the same sort of disorder. Now, the expansion of the repeat basically just gives more places for there to be methylation. More CGG repeats, like what we see here, gives you more places where you have CPG dinucleotides and thus more places where you can add methylation. And Ultimately, this matters because the amount of methylation influences the chromatin state around that mutation. So this is slide 15. The fragility of the X chromosome is a result of highly condensed chromatin in and around that mutation. There are places in your body where, or in your genome where the chromatin is supposed to be condensed. That would be like heterochromatin. However, it's condensed according to sort of I guess like a grammar of the genome that places it in a, heterochromatin is found in areas that are not particularly gene rich, in areas that your body or your cells don't need to have expressed. So there's a kind of an elaborate structure to how that's all set up. If you add sites of constriction or heterochromatin in places they don't belong, then you end up with a rather pathological state, the fragile X chromosome. So this is really just boils down to the interaction between DNA methylation and histone acetylation. They've known since the late 90s that histone acetylation is depleted near the FMR1 promoter in fragile X cells. Histone acetylation, if you can recall the epigenetics lecture, is associated with increased gene expression and then relaxed chromatin state. So the more acetylation you add to the histones, the more the histones tend to repel each other and pull apart the DNA, allowing the chromatin to be open and allowing it to kind of breathe a little bit more. It's a little bit like if you're in bed and you've gotten yourself really tucked in. You're at like a hotel sleeping in a bed for the first night and it's super tucked in. You can kind of like kick around and loosen up the sheets to give yourself more breathing room. This is essentially what histone acetylation is doing. It's loosening up the DNA so that there's more room for it to kind of float around. And this is the opposite of the constriction you see with fragile X. So that's the kind of answer I would have liked to see for that. The answers we got were, pretty good, but didn't quite meet the standard. So when, just as a general rule, I try to give you the number of points I'm expecting for each sub piece of the question. And that of course, I suppose you all would know this in your third or fourth years by now, but that really gives you a hint of where to allocate your efforts. So that's all I have to say about the midterm. Like I said, I'll instruct the TAs to reach out and plan or organize some kind of exam viewing schedule for all of you. I'll have more to say about the final exam in coming weeks as I personally consider it. If you're listening right now, I'll give you a preview. I think it's going to be weighted toward the material on the last third of the course, the last kind of stuff, but it's also going to have a fairly sizable cumulative component because it's worth, it can be worth a fair bit. And I want to make sure that I'm bringing in ideas that come from the whole class, integrating things across multiple parts. So really, I guess if you want to think about how to start studying now, and I'm sure you're all thinking about how you're going to start studying now, I wouldn't even question it, you might want to go back and review some of the earlier lectures where I talk about basic concepts, basic mechanistic genetics and epigenetics, because those things are likely to come back to haunt you in the final exam. I won't say how because I haven't thought of it yet, but that's the kind of thing I probably will think of and ask. So just a little heads up to anybody playing along at home. Okay, so let's talk about major depression. This will be the main mood disorder that we cover today. I think it's, I remember when I was in abnormal psychology for the first time as an undergrad many, many years ago, my professor called it the common cold of mental illness. It's the main disorder in the mood disorders category of the DSM-5. It used to be actually co-localized with bipolar disorder. Major depression was sometimes called unipolar depression, meaning that you're only depressed. Bipolar disorder was, for a while at least, considered to be, two-pole depression, where sometimes you were depressed and other times you were manic. And this back and forth was what separated the two. I don't think anyone really uses the term unipolar depression anymore because it just seems to imply too much connection between bipolar disorder and depression, which psychiatrists, I think, generally believe are separate conditions. Although, to be honest, if you look at the genetics, there's a lot more overlap than you might think. Major depression or major depressive disorder is, as my venerable professor said, the quote-unquote common cold of the world of mental illnesses. Something like 19% of the adult population in the USA has a diagnosis at the time that I prepared these slides. Although, to be honest, I think that the pandemic and recent challenges we've had having to do with the economy and life in general would likely change that number. I think you could potentially argue that as conditions of this sort become more and more prevalent, calling them pathological at all starts to become a little bit strange. You might almost wonder if you have to redefine the parameters of happiness so as to make it so not half of your population is depressed. I know that was kind of a problem with COVID because many people reported higher symptoms of depression than they did prior to the pandemic, and people were already pretty depressed even before the pandemic started. So I guess it's hard to say on a society-wide level, but it's extremely common. And if you think about incidence, most people, I would say, would go through some spell of depression at least once in their lives, although many people have it in a more chronic kind of a way. The average is 19% across the population, but there's quite a bit of variation in subgroups. Females, for example, seem to have about twice the rate of major depression as males. One of the many areas where you see sex differences in the prevalence of a psychiatric condition or a neurological condition. Major depression can appear at any age, but it sort of tends to become common around puberty. Once you hit puberty, life becomes a good deal more complicated, and you end up having to cope with a lot more problems than you would have probably been used to as a child. At the same time, you're going through all these hormonal and neurodevelopmental changes that really make it feel as though the ground is shifting beneath your feet. So it's not really too surprising that major depression appears when you're in puberty. Puberty is around about the time when you first start having serious thoughts, and you start to realize that maybe growing up isn't the greatest thing ever after all. The typical age of onset, however, is maybe more between 20 to 30 years of age, but it can also appear in older people. So 10% of those diagnosed with major depression are 55 or older when they have their first episode. Senior depression is what you would call it. And, hey, being a senior is also a time when your life is changing quite substantially and not often for the better. You are potentially lonelier and less healthier than you've ever been. And again, for those of you who are sitting in your dorm rooms or your basement apartments or your mother's or your parents' houses listening to lectures all day, may seem like things can't get any worse. But you could be doing all those things, but also be really old and feeble at the same time and have growing numbers of health conditions. So, well, that's kind of a pessimistic message, I guess. I mean, there's lots of ways to be healthy and happy as an old person too, but I'm just saying that as a matter of fact, rates of depression do exist among seniors. Why is my text flashing? So the genetics of major depression have interested people a fair bit. What I find interesting about this, I guess in contrast to the other kinds of conditions we've talked about, is that major depression is fairly common, as I said, yet the genetics of it seem to be fairly difficult to suss out. I guess if you think about the way that GWAS studies tend to work, the bigger the number of people you have, the easier it is to find variance. This is a basic fact of the way GWAS works, and you would think that given how common depression is, it would be pretty easy to find. Certainly more easy than it would be to find individuals with schizophrenia. Schizophrenia makes up about 1% of the population. Major depression could be as high as 20% depending on how you define it. So it's a little bit challenging there for some reason. But anyway, twin studies have showed that there's a heritability of about 37%. People who have looked at sex differences in heritability do find that there are differences. However, they're not consistent. And we can look at this over in this forest plot here. So this is a meta-analysis of various twin study-based estimates of the heritability of major depression. And so the heritability in percent likelihood is given on the x-axis, and various studies that they looked at are given here along the y-axis. So where the data are available, they've plotted males and females separately. So for example, this first paper seems to suggest that the heritability is lower for males than it is for females. This second paper seems to suggest the same thing. Heritability is lower for males than females, although the error bars on this are extremely wide, so we don't have a lot of certainty as to what the heritability actually is. Once again, heritability lower for males and females in this study. This study only looked at males, of course, so they couldn't say. Then Kendler et al. Found the exact opposite. They show males as having higher heritability. McGuffin et al. Also show males as having higher heritability than females. Hard to say, folks. It really is tough to say. When you're trying to make these calculations, you have issues of prevalence. If females are twice as likely to have major depression, it can't, like, it's kind of difficult, I guess, to explain that with heritability, because females and males tend to share basically all of their DNA with the exception of the X and Y chromosomes. So females and males are not very genetically different. They do have very different environmental factors. Naturally, females live in a rather different world than males, even in societies that do their best to be equal. There are major differences in the experiences that the two of them have. The dichotomy itself, I suppose you could call into question, although when you're doing these kind of studies, it's inevitable that people want to simplify things down to a basic binary of male and female. The reality is we don't really know why it is that the heritability appears to be different yet inconsistent. I guess it has to do with how you diagnose people, how you categorize the different conditions, and so forth. When you take all these together and collapse across sex, you end up with a conclusion that the heritability is about 37%. That means that the environment plays a pretty sizable role in your likelihood of developing major depression. The unshared environment, that is to say the environment that differs between identical twins, seems to play the largest role in depression incidence. So then there you're thinking about things like stressors and exposure to trauma and various other sorts of lifestyle factors that don't directly relate to your DNA. We can also look at family studies where they find that there's a 2 to 3-fold increase in the lifetime risk of major depression among first-degree relatives, meaning that first-degree relatives are things like parents, siblings, or children, where they're one step away from you, or they're one step removed from you genetically. And this tendency to aggregate is, there's every reason to think this is genetic. There's a genetic cause for this, although, there's always problems in families, and it's difficult to say what fraction of them come from the environment and what fraction come from genes. So I'll give you a brief overview of the history of the genetics of depression, such as I know them. Because depression is such a common condition, yet in a way sort of a mild condition, it's been a little bit more difficult to find really powerful causal variants. For other reasons, similarly, and for reasons that I can't exactly answer, it seems that candidate gene studies have become very popular in studies of depression. I have to imagine this is because depression has, it's common enough that it's a thing, and it's common enough and disruptive enough to people's lives that psychiatrists and people working in mental health have a strong interest in understanding the genetics and are motivated to carry out studies of genetics even when they themselves are not really equipped to do the kind of genome-wide studies you'd really want to be able to do. If you recall, when I was talking about the different ways of learning about genetics, the point of candidate gene studies is that they're fairly limited in the sense that they don't have genome-wide data and thus can be very biased towards giving false positives. But the upside to them is that they're fairly tractable and cheap for smaller laboratories to do. Because they don't require testing across millions of different variants, you can have smaller sample sizes. And you can also conduct more elaborate psychological or behavioral tests. You can have more detailed psychometrics. You can really break down depression into multiple subtypes. You can do structured interviews. You can do a lot more in these kinds of studies because your independent variable, which is your genotype, that's a lot simpler than having a zillion different variants you have to kind of take into account. So the experimental designs can be simpler, and that allows you to study more complicated phenotypes, if that makes any sense. So I'll give you a few candidate gene examples just by way of historical reference. It's, of course, known that many of the drugs used to treat depression involve serotonin and or norepinephrine. The SSRI drugs and the SNRI drugs, for example, are the two sort of frontline drugs that are used to treat these conditions. And for that reason, these drugs, I think, were discovered largely by accident. I don't remember the story of I think that the tricyclic antidepressants were the first ones. And I believe they were originally marketed as like sedatives or something. I can't remember what the story with those were, but hopefully somebody here remembers how it was they were discovered. But anyhow, they weren't being made in order to cure depression, at least at the beginning. But once it was noticed that the drugs that tend to improve symptoms of depression work on the serotonin system, people began to think that serotonin must be somehow involved in depression. For that reason, the focus of many candidate gene studies has been toward genes involved in these known biochemical pathways. So a number of studies have looked at the serotonin transporter gene SLC6A4. This is the serotonin reuptake transporter, the one that is actually targeted by SSRI drugs. And there is a fairly common variant here. It's a 44 base pair repeat polymorphism that's located in the promoter region of the gene. So here we have a map of the gene. This was first mapped out in the late 90s using the kind of old-fashioned mapping technology. So here's the promoter region of the gene. Here's your first exon. Genes, as always, are read from 5' to 3'. And then here you have this 44 base pair indel. So you can either have this repeat inserted or deleted, and the genotype that somebody has is either whether they have the repeat or whether they don't. It's not so much about the length of the repeat, it's just about whether or not they have it. So this is called the 5-HTT-LPR allele, or locus rather. You can have either the long one or the short allele here. The long allele increases the expression of this gene. It's in the promoter region, so it has sort of a regulatory aspect to it. If you have the long allele, you'll have higher levels of expression of the serotonin reuptake transporter in any cells that happen to need this transporter expressed. It won't be expressed in cells where it doesn't belong, but in cells that happen to be serotonergic and participate in serotonergic communication, the expression of this transporter will be higher. And thus you would expect people with a long allele would have increased rate of it would increase the rate of reuptake of serotonin in the synapse. So people can take the genotype here and categorize everyone into long and short, and then measure a whole range of things. So for example, here in the Lesh et al. Paper, this would have been one of the big ones that came out in the late 90s, they look at neuroticism scores for people with a long and short allele. So this is kind of a histogram style plot, and if you kind of follow the contours, you'll see that the peak or the mean for the large or the long allele tends to have a lower score on this neuroticism scale than people who have the short allele, or for homozygous for the short allele. The T-score, I'm not totally sure what that means, but it must be just some kind of measurement of neuroticism. So when this was first discovered, people felt it to be a very promising option, because it's a fairly common polymorphism that actually affects serotonin neurotransmission and seems to be linked to depression. The early studies that tried to make this link, at least, were rather inconclusive. It's not quite as simple as saying that if you have the long allele or the short allele, you'll be more likely to be depressed. It's more a question of people would have hoped for that. Of course, you always want to believe that the simplest answer is going to be the right one, but it didn't turn out to be the case. You can't just go out and measure people's alleles and say, okay, everyone with the short allele is going to be depressed, everyone with a long allele is not going to be depressed, etc. Now, one of the advantages of doing the kind of candidate gene studies is you can iterate upon your hypotheses and test them in more and more refined ways. This isn't really that possible in a GWAS study. With a GWAS study, you're collecting so much data that you really have to plan ahead of time what you're going to look at and then stick to it very, very closely. Actually, I mean, that's part of that is just due to the technical nature of the study design, but also part of it is just ethics. When you have a GWAS study, there's enough variance there, and there's enough things you would measure phenotypically that you could make a whole bunch of different connections, probably on a completely spurious basis. You really have to declare ahead of time what it is you plan to look at, so that reviewers and the scientific community doesn't believe you're just going hunting for data, cherry picking, as they might say, or the old classic Texas marksman analogy. You know, the Texas marksman shoots a bunch of holes on the side of a barn and then paints bullseyes around them after he's after he's already fired. You don't want to do things that way. And apologies to the great state of Texas. Anyway, so but with candidate gene studies, it's not really that's really not the problem. You can kind of do one, see if it works, come back and do another one and see if that works, et cetera, until you find the truth. So there are some real advantages to doing it this way. Anyhow, the early studies to link it to depression weren't conclusive, but people quickly thought, well, what if we looked at moderation by environmental factors such as stress? Because of course, stress and having a stressful life or having multiple instances of trauma or stressful events will raise your odds of mood disorders such as depression. So what if we were to compare that? And sure enough, Caspi and company in 2003 published the first evidence that life stress moderates the relationship between genotype of the 5-HTT-LPR locus and depression. So this is a gene by environment interaction. Something that, to be honest, is all too lacking in our modern, in the modern genome-wide approach to things. You get so much data on the steady state genetics, but no real information about the environment, which I think is a kind of a big deficit here. So here are some plots that they generated. Here on the y-axis, we have self-reported depression symptoms. More means you're more depressed. And then on the x-axis, we have the number of stressful life events. So what you would see in general is that the more stressful life events you've had, the higher your number of self-reported symptoms. This would be, on the Beck Depression Inventory or one of these kinds of psychometric tools. So that's natural, right? Of course, the more events you have in your life that are stressful, the more depression symptoms you're going to report. But the slope of this line was a good deal steeper in people who were homozygous for the short allele compared to people who are homozygous for the long allele. The other thing they measured here was probability of suicidal ideation / attempt. And so again, it wouldn't be too surprising to note that the more stressful events you have in your life, the more the more suicide attempts or the more instances of ideation you might have. That's, I guess, at some level expected. But as with before, the relationship is much stronger in people who are homozygous for the short allele than people who are homozygous for the long allele. So the candidate gene studies, despite their promise, have not always delivered as well as we would have liked. The individual studies, oftentimes within their own context, seem to show positive results or seem to show some kind of connection. However, when you look at this a little bit more closely, you see that on average, they don't tend to show anything that consistent. So here we have a meta-analysis that was done in 2009 by Risch et al. And they're taking just a whole bunch of these studies of the 5-HTT-LPR, spelling mistake there, and looking at their overall effect on depression diagnosis as an odds ratio. So on the x-axis of this forest plot, you have odds ratio, and if you have 1, it means your allele status has no influence at all on whether or not you'll be diagnosed. If your odds ratio is positive, it means that your allele status increases the risk, and if it's negative, it means it decreases the risk. So here are all the studies they've looked at, many times with hundreds, if not thousands, of participants, and the diamond here represents the average of everybody, or of all the studies put together. So you can see that the risk per S allele, or the number of copies of the S allele you have, on average, doesn't have any influence on your risk of depression. When you look at the risk per stressful life event, that does have an effect, where having a stressful life event on its own will increase your odds of depression, typically. This is what this is showing. So a study here is well and truly to the right-hand side of this 1, meaning that it does have a significant increase, or it does significantly increase your odds of being diagnosed. That's true. However, when you put genes and response to stress together, and you're looking at the gene by environment interactions, On average, they amount to nothing. So here the diamond, which is the average of all these studies, is centered exactly on 1, meaning the gene by environment interaction, when you look at it over all the different studies that have been done up to date, doesn't really result in anything. You will get ones like Caspi et al. That show a positive effect and show that your genotype and your environment do interact, but you'll also get ones published later on that have a negative effect, and you'll get ones that have no effect at all. You'll get ones that have a very strong effect but have a small sample size and thus very wide confidence intervals. You really aren't sure what the real, very high margin of error, I guess you could say. You get ones with a very low sample size, very high sample size and thus a very low margin of error. But ultimately, they all kind of disagree and they all kind of cluster around the center point, so that when you put them all together, adjusting for the relative size and confidence you have for each study, you don't get any result after all. So it turns out that this whole thing is probably just a wash. When you put all this together, there's not really any, seemingly not any serious interaction between your genotype and your risk of responding to a stressful life events. Okay, so we have a question in the chat from Ali. So Ali asks, so why do all the candidate gene studies fail? Is it only because of the strong environmental influence on MDD development? I don't think it's so much the environmental influence. I think it's just a sort of an intrinsic flaw of the candidate gene model. If you have millions and millions of different places in your genome where there can be variants and people differ from each other in many, many places along the genome, you can get lucky and pick one that sort of has the appearance of clustering between people. Fundamentally you're asking, are people with depression, does the L or S allele appear more commonly or more frequently in people with depression than people without depression? So that's a single question about a single allele. And if you look at it on its own, you might find that, yeah, sure, more people with depression have this allele, or more, maybe they don't. But if you had picked a different allele that's completely random throughout the genome, your odds of finding the same relationship could also be true, because really you just have to find any allele that differs between depression in your sample, and many alleles would happen to do that purely by chance in your sample. When you do a genome-wide approach, you take this into account and you adjust your significance testing to account for all of the different variants you look at. So you have to have a very, very high standard to say a given allele is associated with depression, because you're taking into account the fact that you're looking at millions of different places. So what I'm saying basically is the candidate gene studies, because they're deliberately only looking in one place, have a high tendency toward false positives. The same way. Let me think here. So the same way, if I were to look at all of the work you had all done in the class and pick a single question out of the exam and look at your grade for that and say, okay, well, that's going to be your grade for the whole semester. That would maybe potentially correlate with your actual grade, but it might not. It's very, very spurious. You can't really just measure one thing out of millions and claim that you found the secret code. Maybe you have. Maybe the biology of it really is appealing. Of course, if the gene is a serotonin transporter gene, it sounds very nice that it would be connected to depression. But ultimately, you're just picking a gene out of a hat, and the odds of you finding relationship are actually pretty high if you just do that. I guess it's kind of what I'm saying. So it's a bit of a false pathway for the literature. It's still being done though. I mean, there is still good work being done somewhat. But I just think as an overall way of discovering the genetics of mental illness, it's not gonna deliver. Okay, so what about pharmacogenetics? People weren't able to show, ultimately I think people kind of abandoned the idea that this 5-HTT LPR genotype had a whole lot to do with your actual odds of depression, or maybe the relationship between your odds of depression and your life events. But what about your response to antidepressant drugs? After all, it does, SLC6A4 codes for the serotonin reuptake transporter, and of course the serotonin reuptake transporter is precisely what's blocked by things like citalopram and fluoxetine and paroxetine and the other kind of SSRI antidepressants. So why couldn't there be an interaction there? And in fact, it would be nice if we found one, because antidepressants have quite variable effects between people. maybe we could use genetics to predict how well somebody would respond to these drugs, and thus maybe find the right drug for them. This would be a terrific thing if we could do this, because getting hooked up with an antidepressant is, as far as I understand, kind of a crapshoot. I think it's, the experts in psychiatry would tell you there's some science to it. I'm sure there is, but anybody who's been through the experience knows that you would try one drug, you go on that for several months to see if it does anything. Maybe, it might do absolutely nothing to you. It might make you have, have a horrible headache. It might make certain parts of your body stop working. It may help your depression. It may not. There's so much variability in the response to these. It would be lovely if we could use genetics to figure out which is the proper drug for somebody. And this is being done. This is being worked on, but the candidate gene approach I don't think really delivers here either. But we do have some consistency at the very least. So here we're looking at the response to SSRI antidepressants. And this is yet again another forest plot, a meta-analysis looking at a handful of studies. And don't worry about these numbers. These are just the samples and everything else like that. This is the weight given to each study based on its sample size and quality. Don't really worry about this or any of this sort of stuff. You can just focus on the forest plot here. And anything to the right-hand side of 1 means the drugs seem to have a more favorable response in people who have the LL or LS genotype. So people who have at least one L allele of this 5-HTT-LPR variant have a more favorable response. And here you do see, even though sometimes the odds ratio or the confidence intervals cross over the middle line, which would typically mean they're not significant, you do see a tremendous consistency here. So all of the studies that have looked have shown that antidepressant or SSRI antidepressants do seem to work better in people who have at least one copy of the L allele. That's what's represented by this diamond here. This is the overall thing. That could be as much of as a twofold improvement in treatment response. So the short allele, of course, is the risk allele for depression. The short allele is the one you don't want. And so not only does it, potentially, I guess we can't totally rule out the possibility that it does actually increase your odds of having depression, or at least having a bad response to life stressors. Not only that, but it does seem to give you a worse response to SSRI drugs, meaning not only are you at a higher likelihood of developing depression, but you're also going to be more resistant to the treatments people tend to use for it. So this, I think, is a fair bit more promising. Now, of course, it still has to be done genome-wide to really make sense of it, and this I think probably could be done. But there is some real promise to the idea that you could use genetics to identify what the best therapeutic options would be for people. And when you have things like antidepressants, which take months to even kick in, anything that could improve your choice of drugs would be a welcome blessing, I would say. So over the years, this course, I've really tried to center it in the genomics era. So you won't see me typically talk about things post maybe 2007 or 2008, because that's sort of when GWAS studies really took off. However, I guess I'm kind of harsh on these candidate gene studies. Lots of people still do them. I mean, they are still being done, even being done locally in Ottawa. And so I don't want to suggest that they're completely useless. But you have to be very aware of their limitations, I guess, before you overinterpret them. And the big problem that we saw in the sort of the pre-genomics era and sort of the early 2000s was that people were very excited to discover the genetic basis of mental illness, and any little hint they found was treated with a great deal of undue reverence and celebration. Over the years, this has gradually faded away. So I just thought I would publish or post here a plot showing the number of papers that come up every year with this search term in PubMed, 5-HTT-LPR. So you can see, starting in 96 when it was first discovered, there were a handful of citations coming out that year. And then we saw this gradual rise to where it peaked kind of in the middle of the Bush administration, or the W. Bush administration, in the mid or sort of early 2000s, and or mid 2000s, I guess. And then since then, a steady decline. And it's still being talked about and still being looked at, but the bloom is a little bit off of it now, and people have kind of moved on. And this would be true of other ones. BDNF, for example, is a commonly, or was at least for a time, a commonly studied candidate gene that's no longer very favored. I guess, if you were to read books about the genetics of this or that, mental health or mental illness and whatnot, things like the Sapolsky books or I don't know, Steven Pinker's books, maybe, books about kind of popular neuroscience books, I think they would give you an oversized impression about the importance of candidate gene studies and what they can really tell us, because they make such wonderful stories. The candidate genes are always, or very often, genes that have a logical connection to the biochemistry of the phenotype you're talking about, in this case serotonin. So it makes for a wonderful kind of thing. And you can look at allele frequency. Another thing that people do that's really kind of not very tasteful is they'll look at the allele frequency of different candidate genes, like oxytocin receptor variations and so on and so forth, and compare them across different races or population groups and say, oh yeah, the reason why this or that country is full of people who behave this way is because they've got more of this allele. You can get into some pretty kind of gross and ugly stuff with this type of approach as well, and people really, really, really take it much further than it's warranted by the actual science. That's me on my little soapbox. So let's get up to the modern era now and look at GWAS approaches. So people have been doing this, and I would say, or I think it's safe to say that, it follows the same basic rules of other GWAS studies, and that's kind of why I've spent a lot more time talking about candidate genes, even though they're not really super, they're not really a super big part of the way the work is done now, is because you've seen enough GWAS studies to know what they're all about, and I don't really have a huge amount more to say about them. It's been done. 2018, I think, is the most recent one that came out for major depression, although there's typically one every couple of years as they add more and more samples to it. But this is a genome-wide meta-analysis, so it takes into account data from many, many different GWAS studies and kind of smushes it all together. That's the beauty of GWAS, by the way, is you can just keep adding more and more data onto the pile and just making your results more and more powerful. But there was over 130,000 cases and over 300,000 controls, so very nice big sample size. And after all that, they identified 44 variants that were significantly associated with depression. So that's all very kind of normal and within our expected parameters for this kind of thing. The thing I will bring up, I guess, is maybe to tell you that the GWAS field isn't quite done yet, although I think the benefits are beginning to kind of trail off. So here is a plot given by the authors of this paper, where on the x-axis they have the number of cohorts that they've studied, and the sample size overall is given by these numbers. And on the y-axis they have the number of variants that they find significantly associated with depression. And basically what it's showing is that as your sample size increases, the number of depression-associated variants you find increases as well. And if you kind of follow this, maybe it's plateauing, maybe it's still going up, but the thinking would be that if you could add more and more samples to this study, you'd be able to find more and more variants up to a point where you could explain all of somebody's risk of depression if you just could finally get enough samples to increase the statistical power. So that's a thought. You do see, when you look at these Manhattan plots, points that are just kind of close to the cutoff line for significance, but just not quite there. If you added maybe another 50,000 people, those points would probably come up above the line, and you'd be able to say you discovered some new regions that are associated with major depression. So there is hope that we would find even more, although it's getting a bit ridiculous. Honestly, hundreds of thousands of people you're going to have to genotype. It gets to be a little bit absurd at this level, but this is kind of, I think people, for all the busting I've done on candidate gene studies, GWAS studies have kind of seemed like they're sort of starting to run out of steam as well, because you can only make these samples so big. Now, if we look at the results from this is another common pattern you'll understand from this course by now. You have these variants that you have linked to major depression. They're all located in genes you may never have heard of. They're not going to be all your nice serotonin genes or BDNF or genes that you might be familiar with. They're going to be in weird random parts of the genome that you wouldn't necessarily be too familiar with. So, as always, you must resort to computational tools to make sense of this. So here, the authors used data from the GTEx set, the genotype tissue expression database, which just measures gene expression in various tissues of the human body and tries to correlate it with genetic variants to find eQTLs. And they were looking at different tissue types and what genes they express, and seeing if genes from the GWAS study were enriched in any of these tissue types. And maybe it's a little tough to see here, but you'll be able to look at this on the slides. But the top hits they found were all brain related. So you've got frontal cortex, cortex in general, anterior cingulate cortex, putamen, which is part of the basal ganglia, nucleus accumbens, also part of the sort of basal ganglia striatal complex, hippocampus, substantia nigra, caudate nucleus, amygdala, cerebellum, hypothalamus, and so on and so forth. The cutoff line for p-value or for statistical significance is here, so really you're just talking about mostly just kind of cortical and striatal areas as being associated. And maybe perhaps not surprising at all, depression genes are not expressed in the pancreas. That's the last one on the list. We could kind of bring this over to, we could bring this to Galen or to Hippocrates and say, listen, we've got genetic proof that you don't think with your liver or with your heart, you think with your brain, and that's where your mood comes from. It's kind of funny, I guess, in a way, like this is sort of a trivial point, right? Obviously, depression genes are probably going to be brain genes or genes that are expressed in the brain that almost doesn't even require a figure to make, or it doesn't require you to even do an analysis, although you still have to. But it's kind of neat, in a way, it's sort of a super modern extension of the very basic question that early anatomists had to ask, which is, where does the mind live? Does it live in the heart? Does it live in the blood? Does it live in the brain? And if it's in the brain, does it live in the ventricles or does it live in the gray matter? Where is the mind located? Where is the soul located? Where does the human experience, where does the subjective human experience actually emanate from? This is a question that's been asked many, many times. And I think it is interesting to look at the different ways you can answer it. There are many, many ways to do it, but this figure kind of represents one of the most modern ways of answering it. If you look for the genetics of depression, you'll find that those genes tend to be all brain expressed genes. Think about trying to explain that to Hippocrates. I'm sure if he could come to our class, he would learn it. He probably would get the best score on the whole test if he just had a chance to learn. But anyway, it's amazing how much of knowledge is built up on previous antecedents. So the old boy would need quite a bit of catch up. Anyway, so if you ask about cell types that are involved in depression or that express depression-related genes, unsurprisingly, we have neurons as the top hit. So this is, I think, a little bit controversial because you could break this down into neuron subtypes, excitatory versus inhibitory, for example, projected neurons versus interneurons, these kinds of things. But low expression of oligodendrocyte or astrocyte genes. So it seems as though, at least on this basis, genes that are genetically associated or genes that come up as GWAS hits for depression seem to be mostly neuron genes expressed in areas of the cortex and the striatum. That's what we can say there. We can look at genetic correlations with major depression. And you all know how to understand correlations now. You wouldn't need to read every aspect of this table. And of course, I won't test you on any one of these particular numbers, but I do want you to understand, well, I already asked you about this on the exam, so clearly I know if you understand it or not. But I want you to really appreciate what I mean by genetic correlations because it's become kind of a standard thing to do in GWAS studies. The authors of this paper looked at the genetic correlations between major depression and various other phenotypes. And probably not surprisingly, the phenotypes that were most genetically correlated with major depression, as measured by the authors here, were just other measures of depression. So you've got the charge scale or the SSGAC scale. Those were very high correlations. The R, this number here is what actually tells you the correlation coefficient. So 0.91 and 0.98 are extremely high correlations, almost perfect correlations. But you also had positive correlations with things like ADHD, anorexia nervosa, anxiety disorders, very, very high correlation there, 0.8. Once again, saying that anxiety and depression are probably very similar conditions, probably different manifestations of the same underlying neurobiological process. Very comorbid, I should say, as well. You would all, I'm sure you've all heard that in past classes as well. Anxiety and depression tend to be very comorbid. ASD, bipolar, schizophrenia, smoking, and various other things. You can kind of go down the line and look at these. Negative correlations. Obviously, the genetics of depression are negatively correlated with subjective well-being. Not surprisingly, the more depressed you are, the less subjectively well you are, the less you're likely to say, I feel good or I feel great. negatively correlate. Some of the weirder ones here are negatively correlated with age at menarche. This is the age when you first have your first period. Age at first birth. This is for people giving birth. What is the age that they have their first child? Did they have their first child when they were 16? Did they have it when they were 35? That kind of thing. Father's age at death. That's a really strange one. Why would the genetics of depression be negatively correlated with your father's age at death? That would mean that what would it mean? So like the more depressed you are, the younger your father died, I guess. That kind of makes sense, right? The more depressed you are, the younger your father died. Yeah, exactly. So that's a weird one. I don't really know why that would be a thing, but it is. These are all, by the way, just questions for you to ponder. If you're ever thinking about research that you might want to do or things you might want to study on your own time, or just questions to ponder while you're in the shower or riding your bike, try to think of a reason why the genetics of these things would all correlate. It really gives you kind of an insight into what's probably actually happening with human disease in general and human phenotype in general. So yeah, the authors actually in the paper, they go as far as saying that, or as arguing that major depression is probably not a discrete entity at any level of analysis. That's a very strong statement indeed. It's not to say that it's not real, but what they're saying is there's a cluster of things that if you put them together in a certain way, you would describe as major depression. However, if you were to take other parts of that cluster and look at them separately or with other things, it might be anxiety, or it might be bipolar, or it might be other kinds of conditions. So they're arguing that major depression is a category we've imposed upon the world. It's not necessarily something that it has its own independent existence the way like a heart attack does. Interesting things to think about. It's fun to see the way that the kind of philosophy of this intersects with the genetics and the neuroscience of it. So that's all I have to say about depression for now. I just wanted to give you 2 quick slides on anxiety so that we don't leave it out. Anxiety, of course, is the other major type of mood disorder. You can have things like generalized anxiety disorder, panic disorders, social anxiety disorder, as well as specific phobias. These are all listed as anxiety disorders in the DSM. Twin studies also point to a similar level of heritability for anxiety as depression, between 30 to 50%. And various types of family and twin studies do seem to show genetic overlap between the different types of anxiety. What that means is that if for example, if you were to look at one twin, that one twin might be diagnosed with GAD, generalized anxiety disorder. But their other twin may have panic disorder. And those are, as far as the DSM is concerned, separate conditions, but they seem to travel together in the same family. There seems to be just a family predisposition to anxiety in general, and then within every individual person, it may play out as a different kind of version of the anxiety disorder spectrum. And so first-degree relatives of probans, people with anxiety, have an elevated risk for the entire range of disorders. So it's really the case that anxiety runs in families in a way that doesn't distinguish a whole lot between the different conditions. I mean, there are like different prevalences. Obviously, I think phobias are probably less common than generalized anxiety disorder. But within that overall society-wide level of prevalence, there is quite a lot of commonality within families. And anxiety disorders, as I said, are highly comorbid with depression, which is consistent with a documented genetic correlation between measures of anxiety and depression. Makes you think, I guess at some level, that we might even just combine the GWAS studies of anxiety and depression and just put them together and see what happens. That, I think, I kind of mentioned this maybe, when did I bring this up? Maybe in the ASD lecture, is that if you don't have enough samples in your GWAS study, if you have a condition that's highly genetically correlated, you can kind of borrow strength from that. You can almost kind of mash the 2 studies together and say, well, depression and anxiety have an 80, correlation coefficient of 0.8. That's almost, it's a very, very good correlation. Maybe they're close enough that we can just kind of stick them together and see what happens. So you can do that and gain a lot of statistical power that way if you like. So we'll just look at this one Manhattan plot from Purvis et al. These folks published this in 2020. And this is a different kind of Manhattan plot. This is a gene level Manhattan plot, which I'm putting up here just to give you some variety in what you have to look at. These Manhattan plots might be getting a little old for y'all. Here, we're not looking at individual SNPs, we're looking at genes. So essentially what they've done is, like, imagine you've got a gene here like TMEM106B, and it's here, and there's the start site. What they've basically done is they've taken all the SNPs inside of that gene, like these little points are SNPs. They've taken all the SNPs inside that gene, and they've just kind of averaged them all together and said, all these SNPs here, all together in this gene, do they together have any relationship with anxiety? And that's kind of nice in a way, I guess, because it makes the plot a lot simpler to read. There's only 20,000 some odd genes compared to millions and millions of different SNPs you can test, so it gives you a simpler appearance. It also helps you kind of combine, kind of smooth out variation across different SNPs and combine the strength of adjacent SNPs. You know, of course, adjacent SNPs are linked by, are in LD with each other, so they naturally are correlated in the same sort of direction. So that's only kind of a way of presenting it that makes it easier to see. But we can look at the names of the genes, and so we've got TMEM106B is the main one. GAD2, that's glutamate decarboxylase. That's an enzyme that synthesizes GABA. So you've got a GABA-related gene. You've got NTRK2. That's this one here. That's the neurotrophic receptor tyrosine kinase 2. So that's a receptor for not BDNF, but a BDNF kind of neurotrophin. And various others. So I don't want to, belabor this too much because I guess we're sort of out of time, and I think I've said enough about the topic. The basic issues with mood disorders are that the basic issues, I guess, is that while the GWAS studies of major depression and anxiety are growing in complexity, they seem to still lag behind the stronger phenotypes like schizophrenia. I think that kind of makes sense at some level. Depression, it's like that famous U.S. judge said about, I'll know when I see it. Like, you would probably know depression if you had it or if you saw it, but on a bigger scale, when you're talking about hundreds of thousands of participants, it's a little tough to draw the cutoff line between, like, you're really, truly depressed and you're just, like, a really sad person. And when you have that kind of diagnostic fuzziness, it can affect your GWAS studies because maybe the genes only, the strongest genetic relationship only exists for people who are super, super depressed. And maybe for people who are kind of on the borderline and just kind of have the blahs or are just going through a rough patch in their life, maybe they don't have the same genetics. Whereas with schizophrenia and ASD and other such things, it's very obvious when somebody has it or they don't. There's no question about whether somebody has schizophrenia or not once they've been diagnosed. It's pretty obvious. So the sample those samples tend to have a higher discriminative ability, and thus the genetics tend to work out a little bit better. As I said, a number of lines of evidence seem to point to a common genetic factor underlying both major depression and anxiety, and really, mental illnesses as a whole. Now, there is an important need to disentangle the phenotypic and genetic overlap because it's tough to say. I mean, the symptoms of anxiety and depression can overlap a fair bit, and that means that it's sort of natural that their genetics will as well. It's like saying, the genetics of height and weight kind of overlap because your height has a big role in your weight. People who are taller are just heavier by nature because there's more of you vertically. Now, it's not everything. Obviously, weight also depends on your width or your level of adiposity or your level of muscle mass, but height is a pretty big factor for that. So you kind of have to come in and disentangle these different contributions, which is done, but it's tough to be certain how well you've done it. But anyway, I just think it's very interesting to think that these studies all point us toward a kind of a common origin in neurodevelopment as the reason for why mental illnesses exist in general. And I didn't give you any epigenetics today. The reason for this is we've talked a little bit about this in the developmental programming lecture, things like the glucocorticoid receptor in individuals who suffered childhood abuse, for example. I don't personally think that the literature is that informative. Many epigenetic studies have been done about depression, but I don't think that they're very good and really are going to tell you a whole lot about what's really happening. So I don't want to burden you with hearing about it. We'll focus on the genetics for now until the epigenetics improve. But I think it's important that they do improve because, as I said, environment, and in particular unshared environment, is a major factor in your risk of depression. So they're going to have to figure out how the epigenetics of these things work because even in the most optimistic scenario, the genetics only account for 30 to 40% of the causality. The rest of it has to be the environment, and the environment will most likely leave an imprint on your epigenetics. We just have to figure out in what cells, what tissue, and at what time it's happening, and what to do about it. Okay, my friends. So thank you to everyone for your attention today, and like I said, your midterm marks will be up later today once I get around to uploading them. Or I'll give them a final look over to make sure that they're all make sense, and then I'll put them up onto Brightspace for y'all. If you're listening to this, please bear in mind that you should start your assignment very soon because of that thing I told you about the website being slow. You would want to give yourself at least 24 hours from the time you submit your file to when you expect to have the answers done. So I'm just giving you a fair warning now. Oh, we got a question from Ali before we quit. So just to confirm, diagnostic ambiguity is what leads to inconclusive GWAS and candidate study results. I wouldn't say entirely. I think there's also... So diagnostic ambiguity means that you wouldn't necessarily be able to reliably diagnose somebody with true depression versus just some other kind of thing like being sad or having kind of a melancholy personality. That's probably part of it. I mean, I think that's why the large scale studies don't produce such clean results. But I think a bigger part of it could also just be the inter-individual variability. It could just be the case that there are lots of different ways that you could be depressed. One of them, lots of different possibilities, and people vary a lot. Maybe like a way of putting it, I guess, would be to say that it's highly polygenic. Monogenic conditions, one gene in one place will tell you everything you need to know. For highly polygenic things like perhaps depression, lots of people could be depressed and have mutations over here or variants over here. Other people could be depressed and have variants in a completely different place, so that when you average them together, it looks like nothing's happening, but really there's just two separate subgroups that are classified on the basis of having a different sort of genetic background. So that's likely a big part of it as well. And again, another reason why it's hard to do candidate studies compared to genome-wide studies. Hopefully that makes sense. Anyway, so yeah, that's all I wanted to say, folks. And the office hours will not be happening tomorrow, but if you need any help with anything, please contact me, and I'll do my best for you. And we'll be back again on Monday with a talk on personality and also on language, because I've decided that personality on its own is not going to is not quite enough. I want to talk about the genetics of language as well, a basic aspect of human psychology. And yeah, it looks like we're kind of getting toward the end of the semester, folks. So just keep it up for the last couple of things that you need to do for this course. Resolve to do your very best on the final exam, and you'll get a very, you'll have a very nice benefit from this in terms of how the grades will be weighted. So hopefully that'll be good for everyone. And yeah, we'll hopefully have a good weekend, and we'll see you next time. Bye.